Hello, I'm going to be talking about the topics covered in the second half of cell and molecular biology. In this section, I'm going to be covering the structure of the cell membrane and the purpose of all the membrane proteins and their energy requirements. At the top of the picture, you can see a phospholipid. It's the little circle with the two lines, and you can kind of see a kink. So the top of the phospholipid, also known as the head, is hydrophilic, which is attracted to water, and the tails are actually repelled by water. And this classifies them as hydrophobic. Contrary to popular belief, the phospholipid bilayer is not actually linear. It's actually rounded. So the top layer of the phospholipid bilayer has larger heads, so when they need to connect in order to work, the top heads are larger because they're easier to touch, and when they touch, they create a rainbow shape, almost like a U or a rainbow. And the inner layer actually has smaller heads in order to line up with the larger heads. If it was linear, there would be gaps in between each of the heads, but because of this, there are no gaps. So this picture right here is actually a great picture to help get my point across. As you can see, the outer membrane has larger heads on the lipids, while the inner layer has very, very small heads. So they need to touch in order to work. And in this picture, you can see that every head is touching the one next to it, and that is what causes the circle shape of a cell. So when there's a hole in the membrane, the hydrophobic tails do not want to be around H2O. They pretty much panic when they're around H2O, and they water repel. So the tails will drive themselves to get closer to get away from the water, which will cause the hole to close. So, going back to the picture, you can see there are many different types of membrane proteins. There are integral proteins, peripheral proteins, lipid-bound proteins, channel proteins, glycoproteins, and carrier proteins. The first one I'm going to talk about is integral proteins. You can think of them as almost integrated into the membrane, and they are found all over the membrane. Also, along with the integral proteins, there are peripheral proteins. I'm going to talk about these two together because they're almost like a yin and yang. So, with the integral proteins, you can see that they're really stuck inside of the membrane. And as you can imagine, they're very, very hard to get out. And peripheral proteins are found on the outside of the membrane, whereas the name peripheral, and you can sense that they're pretty easy to remove. The peripheral proteins will actually detach themselves from the cell membrane or even from other proteins. You can also imagine the peripheral proteins as a hormone, and they're just there for one reason. They'll give off a signal and then they'll leave. And peripheral proteins are also found on the inside of the cell. Going back to integral proteins, they're actually classified as a transmembrane protein because they can carry things in and out of the cell. Also, there are different types of diffusion, and there's one called passive diffusion, where certain little molecules can pass through the lipid bilayer and they almost slip through, almost like a ghost. The reason they can slip through is because they're small and they're uncharged. Unlike passive diffusion, where the molecules can easily slip through the membrane, there's something called facilitative diffusion, which requires a door or a passageway to enter the cell or leave. The molecules in this type of diffusion are actually big or charged, or maybe they could be both. But both types of diffusion do not require any energy. In comparison to that, the pump out of the cell does require energy. It needs a lot of energy, and this is called active transport. I'm now going to be moving on to the carrier proteins because I'm going to be talking about active transport. So the definition of active transport is the movement of ions or molecules across the cell membrane into a region of higher concentration, and it requires a lot of energy. There are a lot of different energy sources for active transport, including ATP, light, and kinetic energy. So there are two different types of mechanisms going along with the pump. The first one is antiporter, and the second one is symporter. So going along with the antiporter, there are high levels of hydrogen ions outside of the cell, and the pump will continue to pump them out of the cell. The antiporter will cause calcium to rush into the cell because on the outside of the cell there's a high level of calcium, and with all the hydrogen going outside of the cell, the calcium needs to go somewhere. So the antiporter will force it to go inside of the cell. The symporter is a little more difficult to get the hang of. So in the cell there's a low concentration of hydrogen ions, and the pump will continue to pump them out of the cell, so there will be a high concentration of them outside of the cell. The symporter, inside of the cell, there will be a high concentration of calcium due to the antiporter flooding them in, and the symporter will cause the calcium to move in the same direction as the pump is pushing the hydrogen ions. An easier way to think of it is think of an antiporter. One arrow's going to the left, which is out of the cell, and one arrow's going to the right, which is into the cell, because the antiporter will cause the calcium to move in. With the symporter, think of two arrows pointing left, because the symporter in the pump will pump the hydrogen ions and the calcium out of the cell. Also, I like to think of the mechanism of carrier proteins almost like those rooms where people are decontaminated, where they enter, they'll stand in there and get decontaminated and then leave. This is pretty much what is going on with the carrier protein, but obviously they're not getting decontaminated. So, going back to channel proteins, they pretty much don't use any energy at all. I like to think of them as a guiding or a space to allow the molecules to go through. Going back to facilitative diffusion, this is the mechanism used to get the larger and charged proteins into the cell. In the top right corner of the picture, you probably see the word glycoprotein, and you're probably wondering, what is that? She hasn't even talked about that yet. What a glycoprotein is that it's a protein on the surface of the cell membrane, and it has a short little carbohydrate chain that will protrude out of the surface. What they do is they form hydrogen bonds with water molecules that surround the cell, which will actually help stabilize the membrane structure. Now I'm going to be talking about the formation, docking, and fusion of clathrin-coated vesicles. So I'm going to start off pointing out the receptors. As you can see, they're the gray little hooks at the top of the picture. There's a ligand binding to it, and actually a little ligand will bind to every receptor. The causation of the first pinch, or the clathrin-coated pit at the top left, is because the clathrin started attaching to the adaptin, which bound to the receptors. Once clathrin is bound, it does not stop squeezing. It will continuously squeeze until a little vesicle is formed. In the second picture, you can start to see that the clathrin-coated pit is getting larger, and it's starting to tighten, and you can see the dynamine in the third picture starting to squeeze. While the dynamine is starting to squeeze, you can see that the clathrin does not stop trying to surround the vesicle. Keep in mind that while the clathrin is surrounding the vesicle, it is still constantly squeezing it, and squeezing and squeezing, until the dynamine finally is able to pinch off the vesicle, and the clathrin can fully surround it. After all of that, the vesicle is finally formed. Woohoo! So what happens is, the vesicle will start to get close to the edge of the cell, the membrane. And it doesn't show in the picture, but there is a part of the vesicle called the rab, and there's a V-snare. Both the rab and the V-snare were part of the vesicle from the beginning. They were along the receptors, and once the vesicle was pinched off, they were in business. So the rab protein will come in contact with a tethering protein, which is a part of the cell membrane, and it will interact with rab and almost like anchor on to the rab and start to pull. Also, during all of this, the V-snare, which is part of the vesicle, will come in contact with a T-snare, and they will also interact and anchor onto each other. So both the T-snare and the V-snare and the rab and the tethering proteins will start to pull the vesicle towards the cell membrane. Also, the V-snare and the T-snare, once they are anchored onto each other, will start to twist and then pull the vesicle towards the cell membrane. That's why it's being pulled. And once the vesicle hits the cell wall, it'll connect, almost like fusion. Once the T-snare and V-snare can no longer pull the vesicle, and the vesicle is already starting to fuse to the cell wall, everything inside of the vesicle will start to be pushed outside of the cell. So the ligands that were attached to the receptors will end up outside of the cell where they wanted to be all along. Also, it's important to keep in mind that vesicles do not always go from inside of the cell to outside of the cell. Vesicles can actually travel in between organelles. So in the Golgi body, vesicles are formed to get to each little section of the Golgi. Some vesicles can be formed from other organelles that can be sent to organelles that need the certain thing. I just want to end this section with saying that the overview of RAB interacts with tethering proteins, which is the docking part of the whole thing, while V-snares interact with T-snares, which pull the vesicle toward the cell, which causes fusion. Now, I'm going to be talking about the structure and formation of all of the cytoskeleton fibers. So I wanted to start with that. I chose this picture because it has a great overview of what the structure of each cytoskeletal fiber looks like. You can see the hollowness of the microtubules and how the microfilaments will twist and intertwine together, and how the intermediate filaments almost look like little tubes that will slide against each other. But I'll get into what happens to each one of them, what forms them, and why they're formed the way they are. So I'm going to start with the most difficult out of the three, and that is the microtubules. I just want to start this off with saying that this all occurs at the centrosome, and a microtubule is a hollow cylinder. So I'm going to take your attention to the left side of the picture, and you can see that the little circles come in pairs of two, and that is called a tubulin dimer. So the tubulin dimers usually start off with GTP, and the tubulin dimer has two different sections, the alpha and the beta. So what happens is the GTP tubulin will come in and bind to other GTP tubulin, but over time the GTP will turn to GDP because GDP will be cleaved by GTPase and create GDP and a phosphate. So in the top right corner, the top right, you can see that a microtubule has a lot of blue and yellow tubulin dimers, which is the GTP tubulin dimer, and that is actually the GTP cap. So what happens is GTP will bind to other GTP or GDP, but over time the GTP will start to be cleaved and become GDP. And once it gets so long, all of the tubulin will become GDP, and then it will fray, like shown in the picture below the top picture. Looking back to the top right of the picture, you can see a lot of GTP is being added to the microtubule. They will constantly be added, and while the microtubule grows, they will create seams like seen in the top left, so the microtubule will become one cylinder. Also, I forgot to add, in the microtubules, the plus end actually grows faster than the minus end. So here's a picture of a microfilament, which is also known as an actin filament. So a microfilament is composed of two strands of subunits of actin, which is why it's also called actin filaments, wound up in a spiral. To be a bit more specific, the actin subunits that come together to form the microfilament are called globular actin, or G-actin, and once they're joined together, they're called F-actin, or filamentous actin. Sorry, I have a hard time saying that word. Also, just like microtubules, the plus end of the microfilament grows faster than the minus end. So, the microfilament will begin to form when three of the G-actin proteins come together to form a trimer. The process of this self-assembly is actually aided by autoclampin proteins, and they act as the motors to help assemble the long strands. So on the picture, you can see the word tropomyosin in this green weaving-like pattern wrapping around the D-actin filament. That's tropomyosin, and tropomyosin actually wraps around the actin to prevent myosin from grabbing it. In the picture, you also see troponin, a blue blob-like figure on top of the tropomyosin. What troponin does is it attaches to the tropomyosin and it lies within the groove between actin filaments in muscle tissue. For example, in a relaxed muscle, tropomyosin will block the attachment site for a myosin crossbridge, which will actually prevent contraction. So I'm going to be moving on to the final cytoskeletal fiber, and this is an intermediate filament. So to start with the intermediate filament, you start off with a single polypeptide. You can see the head and the tail in the central domain. Then that polypeptide will join with another and create a dimer, and they almost weave together, as you can see in the middle. Then, after the dimer is complete, it'll be joined with another dimer and create a tetramer, as you can see in the third row down. And a tetramer are two of the dimers put together, but they're not exactly lined up. After the tetramers are complete, they'll create a protofilament, and it's where the tetramers will actually assemble end to end, as you can see in the fourth row down. After the protofilament is created, 8 protofilaments are taken and interwound to create a filament, as you can see at the bottom. Unlike microtubules and microfilaments, intermediate filaments are stable and nonpolar, which means that it doesn't matter if it's added to the positive or the negative end, or plus or minus. So now I'm going to be talking about the topic of cell crawling, but I really thought this picture was a great example of what cell crawling is, and it brings in the topics we learned last time, or in the last part of the lecture. As you can see, microtubules, intermediate filaments, and microfilaments, but I'm going to be focusing on the microfilaments. I really liked this picture for the topic because it really shows what's happening. So in the top of the picture, you can see a bunch of the actin, which are the red lines, start to bind or create a mesh in the front of the cell. That's called a lamellipodia. On the zoom in of the picture, you can also see these little red channels coming out, and those are called filopodia. And the filopodia are actually formed by the actin bundles being pushed forward, and they will push out of the lamellipodia and create the filopodia. So once the filopodia are out there, they need a way to bind. You can see these little green circles at the end of the filopodia, and those are integrins, which are actually transmembrane proteins that allow for the cell to anchor. Once the cell is anchored, it needs a way to move forward. So what happens is there's myosin motors that bind to the actin, or the actin filaments in the filopodia and in the lamellipodia. It will actually pull them forward. I almost like to think of this whole topic as a little inchworm. It almost moves like an inchworm. It needs a place to anchor, and it needs to be heaved forward in order to move. So now I'm going to be talking about G protein coupled receptors and the difference between using CAMP and calcium as the second degree messenger. The picture may seem quite confusing at first, but I promise I'll talk you through every part. You can ignore the bottom left part of the picture where the three arrows leading to ERK, Rho A, and PI3K. You can see the G protein coupled receptor in the top left of the picture, and you can see a little letter N leading up from the pink cylinder. That's where the receptor is, and things like hormones and other things will land there and will cause a whole reaction. Attached to the G protein coupled receptor is actually the G protein, which can be seen with the bright green circle, and it is made up of an alpha, beta, and gamma subunit. So when nothing's bound to the receptor of the G protein coupled receptor, the G protein remains intact with all three parts, and it's due to GDP. It keeps it together. When a receptor does land on the receptor site, it will send a signal that hits the G protein, and it will release GDP and bind to GTP, which causes it to become active. Once active due to the GTP binding, the alpha subunit of the G protein will almost jump off and it will land on adenylene cyclase. With the addition of the alpha subunit to adenylene cyclase, it will turn it on. And what adenylene cyclase does is that it will catalyze the formation of cyclic AMP from ATP. To be more specific, the adenylene cyclase is actually activated by calmodulin, and then it will start to produce large quantities of cyclic AMP. With all of the cyclic AMP made, it will continue to go around the cell. It will actually diffuse into the cell. For example, it comes in contact with a pKa, or protein kinase A, which is a family of enzymes whose activity is dependent on cellular levels of cyclic AMP. So, protein kinase has several functions, and it includes glycogen, sugar, and lipid metabolism. When the pKa has done its job, it will continue on into this cycle that will actually stick throughout a lot of the topics in the lecture. What happens after is the pKa will send a signal, a kinase will be activated, and to get proteins which will get them phosphorylated, and the cellular behavior will change. When using calcium as the second degree messenger, a lot of it is very similar. When a signal is received, the alpha part of the G protein will hop off, but instead of binding to adenylene cyclase, it'll bind to phospholipase C. Due to the attachment, PIP2 will actually cut a phospholipid into a DAG section, which is the head, and the IP3 section, which is the tail. The IP3, as you can see, is also a second messenger, just like the calcium, but what IP3 does is it goes to a calcium channel in a smooth ER, and it will trigger the gate to open, which allows for a flush of calcium to rush out of the smooth ER. While IP3 is doing that, DAG, which is the head part of the phospholipid, will attach to PKC, which will turn it on, and the turning on of PKC will open a calcium channel, and calcium will rush into the cell. Going back to the effects of IP3, there's a rush of calcium out of the smooth ER, and a protein called calmodulin will be turned on and modulated by calcium, and then the modification will turn on CAM-K, which is a kinase. And going from this is where we repeat the cycle of a kinase is turned on, and then the target proteins are phosphorylated, and then the cellular behavior will change. So now I'm going to be talking about cyclins and CDKs and their roles in cell progression or in the cell cycles. So for all 4 steps in the cell cycle, G1, S phase, G2, and M phase, they all have specific kinases for each step. So what a kinase does is that it adds phosphate groups, or pretty much it phosphorylates. So based on the kinases, there was a study done to see what the protein levels of each kinase was during the cell cycle. And the results were all the kinases were on all the time. They're always going to be there. So they needed to find another way to figure out what exactly is going on. So they did another study, and you can see in the top right corner the results of that study. They're not all the same. They all have different levels. So what you can see is a cyclin-dependent kinase. Or let me start you off with what a cyclin does. So what cyclin does is it ultimately turns on the kinases. So by the cyclin ultimately turning on the kinases, this is what creates cyclin-dependent kinases. So let me walk you through what happens. So during G1, a G1 cyclin will be activated, and the G1 CDK, which is cyclin dependent and knows the G1 cyclin exactly, will only express G1 proteins expressed and activated. So pretty much what happens is because the CDK is turned on, it will cause everything in, let's say, G phase or G1 phase to be phosphorylated, which acts as a marker to see if it can move on into the S phase. If there isn't a marker on one certain part, then the cell has to stay in G1 phase. So going back to the picture in the top right, you can see that there are 3 different peaks, a blue, an orange, and a red peak. And those are all because of the different kinases that will be turned on by cyclin. And the kinases are cyclin dependent because when the cyclin is present, the kinase will turn on. So at the top of the blue peak, you can imagine a CDK, or a cyclin-dependent kinase, is turned on when the protein of G1 is needed. So at the peak of the blue peak, the protein levels are at their highest. Then, when it's not needed any longer, it will drop, and the next CDK will be turned on, as you can see in the orange peak and in the red peak. It's almost like a never-ending up and down and up and down. So now I'm going to be talking about all 5 types of cell junctions. The first type of junction I'm going to be talking about is a tight junction, which you can see in the top left of the picture. So what happens is in a tight junction is it's easy to think about it almost as like a zip or a zipper, and it will zip up and become very tight. But what happens is occludins and claudins will line up next to each other, and they will go, let's say, on the cell on the left, it'll go occludin, claudin, occludin, claudin. And on the right, it'll go the opposite, so claudin, occludin. And once needed, they will zip together and be as tightly put together as possible to create a tight seal. For example, think about like a zipper on a jacket. When the zipper is all the way down, the 2 sides of the zipper are open, so they're not touching. But when the zipper goes up, they'll start to attach and hold together to create a tight seal so nothing can get into your jacket. So now I'm going to be talking about gap junctions, which I personally think is the easiest one of the 5. So in a gap junction, there are 2 channels. So in the top right, you can see that there are gaps between the cells, and there's almost like a channel when both of them come together. There are also connexons, which are composed of connections, and they will help transfer things back and forth between each cell. So think about back to the when a cell is coming in contact, they can go through almost like a channel. That's pretty much what a gap junction is. It's a channel open for signals to be passed, or molecules to be passed back and forth between cells. The next junction I'm going to be talking about is an adherent junction, which is actually for skin cells, which tend to move. So what happens in an adherence junction is you can see on the second row down to all the way to the right, that is an adherence junction. What happens is in one cell there will be coherin and the other will have actin, and they both have a negative charge inside of the cell. So in that filament sticking out inside of the cell, it's both negative, so they tend to keep away from each other. But when a calcium ion, which has 2 positive sides, it will cause it to even out and almost bridge them together and cause them to come together, which creates an anchoring junction, just like seen in the picture. So the next anchoring junction I'm going to talk about is a desmosome, which is involved with the skin. So what makes up a desmosome is similar to adherence junctions. There is a connection to calcium due to two negatives, but instead of actin and coherin, they use intermediate filaments, which I talked about earlier in the lecture. So the intermediate filaments in the desmosome cause for tensile strength, which stops tissues from tearing, and it stops skin from tearing when they're stretched. So think about when you pull on your skin. Why doesn't it rip off? Like, what's causing it not to rip off? Those are the desmosomes keeping in tensile strength in order for you not to rip your skin off, which I'm very grateful for because I would be horrified if that did happen. The last anchoring junction I'm going to be talking about is a hemidesmosome, which is pretty similar to a desmosome, but they're actually completely different, only the names are similar. So in a hemidesmosome, there are also intermediate filaments, but they're actually pointing upward and not to the side, and they're connected to the integrin, which is a transmembrane, and the integrin connects to the collagen below, which also the collagen will attach to the ECM. So it's almost like a whole cycle. And these type of anchoring junctions keep our skin stuck to us. During this whole section, I just wanted to talk about the difference between apical and basal surfaces, which has to do with the cell. So an apical is the top of the cell, and the basal is the bottom, where it's anchored. And it's anchored to the basal lamina, which in the case of the hemidesmosome is collagen. Also, the membrane domains, which can be found in the top corners and the bottom corners of the cell, can laterally diffuse, but they cannot diffuse downward. So I'm going to end this video talking about how cancer is a sociopathic cellular condition. So most of the time, the body's cells will work together in harmony and they all contribute to the greater good. Cancer cells, which will rapidly divide and colonize the body, are actually an exception to this. The reason they prosper is at the expense of other cells, but their final product, they can eventually kill the host, which can be people. Usually, when the cell begins to go wrong, like if something's damaged, they get signals that tell them to self-destruct. But mutations acquired can cause the cell to resist these signals. Once the changes encourage it to start dividing, it actually becomes a cancer cell. So eventually, after a whole bunch of cancer cells or a tumor will build up, and it'll stop growing because oxygen can no longer get to the center of the tumor. But that is where, all the way to the right of the picture, you can see that the blood vessel will be brought to the cancer cell. It acts as a way for the cancer cell to continue on growing and spreading. And once it gets into the bloodstream, that's where the cancer will become metastatic, which is probably the worst type of cancer possible. So all in all, by getting all the genetic changes, cancer cells will acquire different properties that enable them to overcome all different types of body defenses. They get an advantage, and the number of spreading will increase. Also, sometimes tumors can become resistant to different drugs used, but that's a whole different story. So I just wanted to bring attention to the size of the different types of cells, the normal cells and the abnormal cells. You can see that the normal cells are pretty small, and the cancer cells are huge. They're almost like a supercell. And this is very important because supercells can easily just manipulate and bully almost normal cells. And that's why cancer is so dangerous. So all of what I just talked about really shows why cancer is a sociopathic cellular condition. It pretty much only thinks about itself and what it can do to continue to grow and divide and even invade. Cancer cells pretty much do what they need to survive. They'll get rid of any other potential harm to the cell, and they'll just continue to grow. And if they continue to grow, it's harder to take them down pretty much. Also, they bully all the other cells, and they are seen as number one. They always want to be seen as the most important. So I just wanted to end this section off with probably one of my favorite quotes Dr. Manella has ever said in the class. It's not word for word, but this is how I really interpreted it. Cancer is a disease of accumulated mutations, because pretty much that is what it is. A lot of different mutations will cause the cancer cell to start rapidly dividing, and then the mutations will cause them to start invading, and then they could start moving throughout the body. So pretty much cancer is just a bunch of mutations, and it's very, very hard to stop. And it is very hard to prevent. So this marks the end of my entire eReport for the second unit of cell and molecular biology. And I just wanted to thank you for watching and spending your time learning about all I learned in this half of the course. Once again, I wanted to thank you for watching and have a great day.